Hello, everyone. My name is Michael Story. I'm the Vice Chair of the Department of Radiation Oncology, Chief of the Division of Molecular Radiation Oncology, and the Director of the university's Preclinical Radiation Core Facility. Before I talk to you about the subject of the slides, I wanted to give you a little introduction to the Division of Molecular Radiation Oncology. We're roughly 90 individuals, including 12 principal investigators, probably another 20 research faculty, and then postdocs, graduate students, technicians, and administrative staff. We have a very broad research portfolio. Some of our principal investigators are considered world experts in the area of DNA repair, from mismatch repair to non-homologous end joining to homologous recombination. In addition, we have multiple faculty members who have backgrounds in immunology, and as you can imagine, their research portfolio looks at a combination of radiation with immune checkpoint inhibitors in order to enhance radiation therapy. Our group also has a long-standing research portfolio for NASA, specifically in the area of radiation-induced carcinogenesis or cardiac dysfunction, and even cognitive issues. My lab, for instance, has been funded for over 20 years by NASA, looking at carcinogenic effects from these heavy charged particles that you find in free space. As a consequence of understanding or trying to understand the biology of these heavy particles, several of us have a keen interest in using heavy charged particles for radiation therapy as well. If you're an incoming resident, you should be aware that a number of our residents have done their research in the labs within our division. We are quite amenable to Holman applications. We have one Holman fellow in my lab right now, and we're working on the applications of a couple more for the coming year. I would encourage you to look at our website and see the activities of our labs, and if you find that there's anything interesting for you, please contact the respective faculty member. So what I'm going to talk to you about in this slide deck is something you may not have heard of. It's called tumor treating fields. It's a relatively new cancer therapy, and I'm going to discuss our research for the last few years in this area. Tumor treating fields are essentially low intensity, intermediate frequency, alternating electric fields. You can see here this gentleman is being treated for glioblastoma, and he has an array of ceramic transducers that have been placed on his scalp in the region surrounding the tumor. You see this cord draped over his shoulder that goes down into a battery pack. And this battery pack controls the application of these electric fields in a perpendicular fashion. Tumor treating fields were approved by the FDA for recurrent and newly diagnosed glioblastoma, as well as now being considered as a compassionate use device for mesothelioma. The FDA approval is in part based upon the results of this phase 3 trial, EF14, a multi-center trial where you can see both progression-free survival and overall survival are enhanced by the application of tumor treating fields um, compared with temozolomide compared to temozolomide alone. Well, a number of years ago, representatives from the company that devised this technology came to us to see if we were interested in their product and in determining whether the mechanism of action that they've described is the only mechanism of action. So it was clear from what they showed us that the main effect at that time for tumor treating fields was through an antimitotic mechanism of action. And essentially, if you apply tumor treating fields to cancer cells, because of the electric field, you set up dipole moments for polar molecules. And you can see, in this case, this is tubulin. They align based upon their charge and their exposure to the electric field. And this disrupts the production of microtubules. The result is mitotic delay, possibly death in mitosis, abnormal divisions, perhaps even slippage so that cells wind up after division with both nuclei instead of one. Abnormal mitoses can result in death by interphase. And if cells escape this, they are unfortunate enough to have to go back around and go through this again. You can see disrupted mitoses here in the left for non-small cell lung cancers, ovarian cancer cells, and here's an example of ovarian tumor cells either treated with tumor treating fields or not. You can see there are plenty of cells growing on this dish while there are none on the dishes that were exposed to tumor treating fields. Now, they also had fluorescent microscopy, live cell imaging, and you can follow what normally happens to a cell in mitosis. You can see this cell divided in roughly 49 minutes, and with the help of a little bit of an illustrator, you can see the ordinary separation of the chromosomes and subsequent cytokinesis. Now, in cells exposed to tumor treating fields, if you watch these cells, it takes a significant amount of time for them to undergo mitosis. They have not properly wound up. They have issues with developing their spindle, and we will go several hours actually before this cell finally commits suicide, essentially, by mitotic cell death as we switch to phase contrast. Well, we had no issue with their mechanism. It certainly, the data they showed us, it seemed a reasonable approach, but in my mind, it did not explain the amount of cell death that they were describing, at least in their in vitro experiments. And as you might imagine, the numbers didn't align in that mitosis is a very small part of the cell cycle. Maybe 5%, 2% of cells in a tumor could be in a mitotic phase. Clearly, the opportunity for effects based upon these electric fields and the potential for disruption of structures that require polar molecules suggested to me that there was an opportunity to look in interphase to determine whether there were other mechanisms that might explain the enhanced cell death that we were seeing. So at the time, my laboratory was very interested lung cancer, and they were starting a trial called Lunar, which was the application of tumor treating fields to lung cancer patients. And so we suggested to them that we are very interested from the perspective, though, of the application of tumor treating fields and radiation. And because we had a large portfolio of lung cancer cell lines, we would also look to see the consistency of the effects of tumor treating fields across a broad array of lung cancer cell lines. So what you see in this figure are 5 lung cancer cell lines, and they actually vary in their response to tumor treating fields. The 2 at the top are much more responsive to tumor treating fields than the 3 cell lines at the bottom. And in fact, when you irradiate and then apply tumor treating fields, the effects were actually synergistic. And we published this and other data in 2017. So because we have this enhanced radiation and tumor treating fields effect, we wanted to know whether that actually would manifest itself in increased DNA damage, or perhaps increased an inability to repair the damage that is generated by radiation. So you see these 3 panels here. The top panel is looking at overlapping 53 BP1 and gamma H2X proteins. The foci of those proteins is a function of time after radiation exposure. And you can see this clear difference in the repair of these DNA double-strand breaks when tumor treating fields have been applied, which is the dashed lines. So you see that the rate of repair has been slowed. This is 2 of the more permissive lines, 2 of the more resistant lines to tumor treating fields. Interestingly, though, if you just look at gamma H2AX, which is more than just a metric for DNA double-strand breaks, it's also a metric for replication stress. You can see that the effect is far more profound. But if you look at the blue lines, this is what we found to be quite interesting, and that is that if you apply tumor treating fields alone, over time, you see an increase in the production of DNA double-strand breaks. And these values are quantitated here below, and I won't go into it other than to say that they are statistically significant differences. Now, these pairs of blue lines and the fact that gamma H2X may be an indicator of replication stress, this made us think that what we probably need to do here, because we have these cell lines that respond versus those that do not respond, was to go ahead and do a genomic analysis and see if we can segregate the responses of these cell lines based upon their response to tumor treating fields. And then by having essentially a transcript analysis, we could better define how this, what the mechanism of TT fields was. So as you can see here in our heat map here, it was quite simple actually to segregate these 5 cell lines based upon their response to tumor treating fields, even as a function of time of exposure of tumor treating fields. And given the fact that we have reduction in DNA repair capacity, we have the potential for increased replication stress. In fact, the #1 pathway that we saw altered in the more responsive cell lines was actually the role of BRCA1 in the DNA damage response. If you look at the cell lines that are less responsive, the role of BRCA1 shows up as the third most differentially regulated um, pathway, and the fact that these are both blue suggests that these pathways are downregulated. Well, BRCA1 is actually part of the Fanconi's family of DNA repair genes, and so we looked at the protein level as well, and indeed you can see when we apply tumor treating fields, we see a reduction in the protein, so we have a reduction in the message as well as the protein, and that also held true for FANC-D2 across these cell lines as well, and FANC-A, and as it turns out, a whole host of other genes that are associated with the FANC pathway genes were shown to be downregulated. Well, while the Fanconi's genes didn't necessarily explain their radio response, there were a number of other pathways associated with DNA repair and cell cycle regulation that were also downregulated. However, a number of members of the Fanconi's pathway genes are associated with replication stress. So we decided that let's have a look at replication stress, and we did that in two ways. One was we looked at newly replicated DNA. Essentially, the length of that DNA is a function of time when cells were exposed or not to tumor treating fields. And you can see in the two panels here that, in fact, as a function of time, the length of newly replicated DNA, which is demarcated with the red lines, is actually left shifted, suggesting that indeed the replication forks are stalling, slowing, perhaps even collapsed. Then we looked at something called an R loop, which is a DNA-RNA hybrid structure that's generated when you have collisions between DNA repair and the replication machinery, and you have these single strands of DNA and RNA that can combine. And so you can see as a function of time for H1299 and H157, resistant and permissive to tumor treating fields, you can see an increase in the production of R loops, which is determined simply by the antibody attaching to this DNA-RNA hybrid structure, the S9.6 antibody, which is a very common antibody used in genomic analysis, actually. And for the 157 cell line, the more T2 field's permissive, you can see a large increase in the production of these R loops, suggesting that indeed these cells are under significant replication stress. So if you look at this diagram here that we took from Zhang et al. Cell reports in 2016 and have modified a bit, where they're essentially looking at the replication stress level, we argued that tumor treating fields are essentially pushing the extent of replication stress from low to intermediate to high, and that the events that are associated with replication stress, like fork stalling, fork collapse, etc., can be taken advantage of by either using chemicals that create DNA damage, like including radiation, because you have a reduction in the genes associated with DNA repair or cell cycle checkpoint. But also you could intercede here in any point along this pathway where you might inhibit the the maintenance of replication forks, which is a significant job of the Fanconi's pathway genes. And you may also cause concerns for chromosomal integrity, chromosomal maintenance. So the argument is that with the downregulation of DNA repair cycle checkpoint DNA replication fork maintenance genes and proteins by tumor treating fields, you cause increased replication stress, rendering cells susceptible to DNA damaging agents, DNA repair inhibitors, and inhibitors of replication fork integrity. So just to give you some examples of our test of that notion, here's an example of the combination of the tumor treating fields with cisplatin, and everywhere you see these asterisks, the effects were synergistic with the application of tumor treating fields. And here on the right, an example of two cell lines where we used a PARP inhibitor with and without radiation. And again, the effects are synergistic, and particularly when combined with radiation. And we have a patent application on the use of PARP. Interestingly, PARP is one of the proteins that is actually upregulated when cells are exposed to tumor treating fields, which actually makes sense. PARP is being upregulated in its role for replication fork maintenance, while other proteins associated with replication fork maintenance are downregulated, which actually causes the upregulation in PARP. So using a PARP inhibitor just makes life worse. This is also shown when you have the combination of cisplatin and etoposide, and here we're considering the implications for using tumor treating fields for lung metastasis to the brain. Clearly, tumor treating fields are used for GBM, and here's the potential for using agents like cisplatin and etoposide in combination with radiation to treat brain metastases. So, a couple of very complicated diagrams, but we'll walk through it slowly. You start out with dividing tumor cells, and you apply tumor treating fields, and you can have disrupted mitoses, and essentially walk down this pathway of cell death, slippage, or coming back around and going through the process again. You can have dividing cells, apply tumor treating fields, and then use radiation, UV light, chemicals, whatever, cause DNA damage, and tumor treating fields block that DNA damage, and you have failed DNA repair, you have cell death. It would also appear that you may also, because you have fragmented cytosolic DNA, that you may trigger innate immunity and get an immunogenic response resulting in enhanced cell death. But what our data showed, frankly, is that for cells in interphase, if you use extended treatment with tumor treating fields, you see the downregulation of the FANC genes, disrupted DNA replication and repair machinery, replication fork stalling. The result of that is replication errors, collapse of replication forks, development of R loops, and single-strand and double-stranded DNA, which that can result in cell death or innate immunity. But then if you apply radiation or other chemicals subsequent, you see an enhanced cell-killing effect. So tumor treating fields obviously cause an antimitotic effects. They cause DNA damage and replication stress. They cause immunogenic cell death. Data we haven't shown you is in autophagy. We show actually that tumor treating fields block mitophagy. And some recent results suggest that tumor treating fields actually open up the blood-brain barrier, which may allow for the use of chemo agents that ordinarily would not cross the blood-brain barrier. Okay, so that's all well and good, but what's driving this dysregulation of DNA repair, etc.? Well, we thought that genomics data is probably not identifying the key events, as they could be post-translational, or they could be early transcription events. So we decided to do a proteomic analysis, and we essentially use a tandem mass tag labeling to tag proteins in cells exposed to tumor treating fields or not. And we ultimately were able to quantify about 7,500 proteins at an false discovery rate of less than 1%. And interestingly, a large number of proteins are actually downregulated when exposed to tumor treating fields, while there is a modest number that are also upregulated. You can see here in the panel on the right, again, we can clearly segregate the treatment of cells with tumor treating fields based upon their protein expression. Now, we use the string database to look at the interaction networks, and what we found was a similar profile as we saw with gene expression, in that we saw DNA replication and damage pathways altered. We saw mitochondrial dysfunction. In particular, we saw changes in ubiquitination that are associated with mitophagy. And we saw some of the cell cycle pathways also altered in their regulation. But we also saw translation and transcription pathway changes. So in our interrogation of those pathways, we hit upon the fact that we were seeing dysregulation of the E2F family of transcription factors. Now, there are a number of E2F family members. We have 3, 6, 8. 3 of them are activators, while as 5 of them are actually repressors of transcription. So in the activators, E2F1 and E2F3, we saw them downregulated. And in the repressor family, particularly E2F4, and although not here E2F6, we saw those upregulated. So to reiterate, the activators of transcription were downregulated, the repressors of transcription were upregulated. And you can see here the genes that are regulated by this transcription factor family and their expression ratios. Now, there are, of course, a number of target genes for the E2F family members, and some of them are listed here, but for Fanconi's anemia, you can see BRCA1, FANCD2, MLH1, and others associated with chromosome maintenance, and of course mitosis and cell cycle regulatory genes as well, and DNA replication targets. So we were looking around the literature and decided that a novel approach to using tumor treating fields may be to target the E2F/RB CDK4/6 axis. And there are agents that are either approved or in clinical trials already. For instance, there's an E2F inhibitor, HLM6474. It's a kind of a pan E2F inhibitor, and it's in lung clinical trials and has been shown to synergize with paclitaxel. And of course, there are a myriad CDK 4/6 inhibitors, but we chose this LY 2835-219, which is actually FDA approved for primary and recurrent brain tumors and metastatic breast cancer. The idea here is the application of tumor treating fields would result in a dysregulation of the E2F family members. We would also then apply the CDK4/6 inhibitors, and we would essentially wind up dysregulating this entire stream of E2F family member activities, and that the target genes would include cell cycle, the Fanconi's genes, DNA damage and replication, the result being abnormal cell division, DNA damage and replication stress, leading to abnormal cells and ultimately an enhanced cell death. And indeed, although this is a very, very complicated graph with two cell lines, just looking at the application of the E2F or the CDK4/6 inhibitors or the combination of these inhibitors with or without tumor treating fields, we saw the greatest synergy of any combination that we have ever used, whether drug or radiation, when we combined these two agents. So generally we would see values, so-called confidence index values of 1 to 3, maybe 4 with some of the other agents. Anything greater than 1 is considered synergy, but we're seeing values of 8 or 11, for instance, with the combination of these agents. So to summarize, tumor treating fields induce replication stress through the downregulation of the E2F transcription factor family. That allows us to target the CDK4/6 E2F-RB axis, which in and of itself dysregulates a variety of pathways, including DNA repair, cell cycle, chromosome maintenance, DNA replication, and replication fork maintenance. The result being a substantial increase in cell death. Essentially, tumor treating field exposure leaves tumor cells vulnerable to DNA damaging agents or inhibitors of the above, leading to enhanced cell killing. Tumor treating fields increase the sensitivity of cisplatin etoposide, which can be tested in patients with brain mets, for instance, from non-small cell lung cancer. And it also provides us with new clinical strategies. For instance, tumor treating fields should be used concurrently with radiation. Tumor treating fields could be used in the interim between diagnosis and surgery, for instance, to reduce tumor bulk with little effect on the patient. Tumor treating fields could be used in tandem with immune oncology. But most importantly is that there's now a mechanistic rationale for tumor treating fields in drug therapy and a mechanistic rationale for tumor treating fields in chemoradiation. So with that, I'll stop this presentation, and I look forward to talking with any of you. Please go have a look at the division webpage and my lab's webpage. You can find more on tumor treating fields. And if you have any questions, you can reach out to me. I'm happy to answer any questions you may have.